In this section, we will talk about the frameshift mutation. In the early 1960s, Sidney Brenner argued on theoretical grounds that the code had to be a triplet, since three-letter words represent the minimal use of 4 letters to specify 20 amino acids, and a codon with 2 nucleotides would not yield enough combinations to code for the 20 different amino acids that commonly occur in proteins, because 4 into 4 equals 216, but because a codon with 3 nucleotides would yield enough combinations to code for the 20 different amino acids that commonly occur in proteins, because 4 into 4 into 4 equals 264. That's why a codon with 3 nucleotides would yield enough combinations to code for the 20 different amino acids that commonly occur in proteins. Experimental evidence supporting the triplet nature of the code was subsequently derived from research by Francis Crick and his colleagues using phage T4. They studied frameshift mutations, which result from the addition of 1 or more nucleotides within a gene, or deletion of 1 or more nucleotides within a gene, and subsequently the messenger ribonucleic acid transcribed by it. The gain or loss of letters shifts the frame of reading during translation. Crick and his colleagues found that the gain or loss of 1 or 2 nucleotides caused a frameshift mutation, but when 3 nucleotides were involved, for example, cytosine, adenine, guanine, the frame of reading was re-established. This would not occur if the code was thing other than a triplet. This work also suggested that most triplet codes are not blank, but rather encode amino acids supporting the concept of a degenerate code. Let's understand frameshift mutations on a deoxyribonucleic acid sequence with the repeating triplet sequence: guanine, adenine, guanine. This is an initial frame. It has triplet sequence: guanine, adenine, guanine, such as guanine, adenine, guanine, guanine, adenine, guanine, guanine, adenine, guanine, guanine, adenine, guanine, and guanine, adenine, guanine. The insertion of a single nucleotide, such as cytosine, takes place. Now, after the addition of single nucleotide, which is cytosine, new sequence shifts all subsequent triplet reading frames, and all become out of frame. It means the gain or loss of 1 or 2 nucleotides caused a frameshift mutation, but the insertion of 3 nucleotides changes only 2 triplets. But the frame of reading is then re-established to the original sequence. Let's understand it. This is initial frame, addition of 3 nucleotide, such as cytosine, adenine, guanine. A new sequence will be produced. The insertion of 3 nucleotides changes only 2 triplets, and they become out of frame. But the frame of reading is then re-established to the original sequence, and they come back to frame. This was all about this section. In our next section, Dr. Aliana will join us, and we will talk about the coding dictionary.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.